Okay, this is podcast number 2 for Standard 2.1b, which is going to be all about the different types of transport that can happen through the cell membrane. So on this slide, there actually is nothing for you to write. I'm just going to do a quick overview of the two types of cell transport, and then we'll get into the notes in the next slide. So basically, we have two types of transport that can happen in a cell. There's passive transport. In passive transport, the cell is not going to use any energy. So we're talking about diffusion, facilitated diffusion, and osmosis. So you can imagine that like being at the top of the hill, and you got to get to the bottom of the hill, and you're on your rollerblades, and you just go right down. Doesn't take a whole lot of energy. Active transport is the opposite of this, where the cell has to use energy in order to transport molecules across the membrane. Protein pumps, endocytosis, and exocytosis are all examples. And so you can imagine that as being at the bottom of the hill with your rollerblades on, and you need to get to the top. It's going to take quite a bit of energy and effort on your part. So getting into these notes, let's talk about what passive transport is. As I just said, the cell is going to use no energy for passive transport. If you think about what passive means, if you're being passive, you're just kind of like sitting back and relaxing. You're not really using any energy. The key to passive transport is that molecules move randomly, and molecules are always in constant motion. And so their goal is to have as much room as possible to do this random movement. And so with passive transport, they are moving themselves around so they can get as much room as possible to move around. Molecules in passive transport are going to move with the concentration gradient. What this means is that they are going to move from an area of high concentration to an area of low concentration. So they're going to go from where there is a lot of them down to where there is fewer of them. You can think about this in terms of if somebody sprays perfume on one side of the room, the people that are sitting nearest to where that perfume was sprayed will smell it first, and the ones that are further away will smell it later because it had to diffuse across the room from where it was highly concentrated to where it was less concentrated. Okay, there are three types of passive transport. There is simple diffusion. There is facilitative diffusion. This is diffusion with help. So to facilitate something means to help. So this kind of diffusion is actually going to get help with the use of transport proteins that we had talked about with the cell membrane. There are certain proteins that go all the way through the membrane, so facilitated diffusion is going to happen with the help of those proteins. And then there's a very special kind of diffusion called osmosis. And osmosis is strictly the diffusion of water, and it gets its own category because of the importance of water to living things. So we're going to just quickly talk about each of these in terms, and we'll actually spend a little more time with the osmosis piece. All right, let's start with diffusion. Diffusion is the random movement of particles from an area of high concentration to an area of low concentration. Remember, molecules move randomly and they are in constant motion, so they're going to move to wherever they can get to have the most room to move. So we're moving high to low. So if you look at this is kind of what I was talking about with the perfume. If somebody sprays a perfume over here, it's going to spread out until it's kind of evenly distributed around the room. These molecules are still going to continue to move even after they have spread out, but they are going to get as much room as possible to do that movement. So when we're talking about the cell membrane, only small molecules that have no charge can diffuse through the membrane. So these are molecules like oxygen and carbon dioxide. Now diffusion is going to continue until all the molecules have been evenly spaced out, and this is called equilibrium. So once all the molecules are as spaced out as possible, that is when diffusion will stop. But remember, the molecules are still going to keep moving around, but they're just going to stay spread out. So they're just trying to have as much room as possible to move around. Okay, so here's another example. This is showing diffusion across a membrane. So all the molecules are starting on the inside of the membrane, and then they are moving out, but notice they still keep moving even after they have equally distributed themselves inside and outside of the cell. All right, continuing on with passive transport, the second type of passive transport is facilitated diffusion or facilitative diffusion. So this is diffusion of specific things that have to go through transport proteins that are found in the membrane. So as you can see here is a transport protein in this picture, and so these blue molecules can only get through this membrane by going through that transport protein. The red ones were able to get through the actual phospholipid bilayer on their own, but these blue ones cannot. Probably because they have certain qualities about them that will not allow them to get through the phospholipid bilayer. So they are going to use transport proteins. Now the thing about transport proteins is that they are selective. So only certain molecules can go through certain transport proteins. Just like enzymes are only going to work on certain types of molecules, transport proteins work in the same way. So this transport protein right here may be just allowing glucose in, and there'd be a different one for, say, sodium ions. So depending on what needs to get through, there's a different transport protein for each type of thing. The types of molecules that are going to be transported through facilitated diffusion are usually molecules that are way too big to get through the phospholipid bilayer, so these are larger molecules, or molecules that have a charge, like ions. Remember, our phospholipid bilayer is very selective in that it doesn't really like water in the middle. So anything with a charge is actually going to get repelled away from the membrane. So things that have a very strong charge, like sodium ions and chlorine ions and potassium, they're not going to be able to get through that membrane at all because they get repelled by the phospholipids. Okay. Here's just another example of this moving stuff out going with the concentration gradient high to low. Okay. So if we think about passive transport, here's another diagram of this. So here we've got a bunch of glucose molecules over here, and those glucose molecules need to get to the other side. Okay, so this is cellular transport from a high concentration, glucose molecules are high, to a low concentration, so they're going to go to where there's less of them because they want more room to move. But this has to happen through a transport protein because glucose molecules are too big to get through the membrane on their own, so they have to travel through this molecule here. Be like you trying to pass through the wall to get into my classroom. You can't. You got to come through the door. It'd be too big to go through the wall. Okay. All right. Our third type of passive transport is osmosis. Osmosis is the diffusion of water through a selectively permeable membrane. So we are only talking about water when we talk about osmosis. So water is going to move from high concentrations to low concentrations. So this diagram was going a minute ago, but these little molecules here that look like flies, those are your water molecules, and they are the ones that can move. They can very often move right through the membrane when needed, but they have a hard time with that because remember the tails on the inside don't really like water. They can do it, but it's not easy. So very often there are pores made of proteins, transport proteins, that are also going to help get those water molecules through. So it depends on how much need there is for the water. Okay. Now these solutes that are over here, these green ones, were too big to get across that membrane. Oops. Okay. So why does osmosis get its own category? Well, osmosis has some pretty important effects on life. All right. So remember, osmosis is the diffusion of water through that membrane. Now, water is so small and there is so much of it that the cell oftentimes has difficulty controlling its movement through the cell membrane. And that can cause serious problems for a cell. So if you have a cell that is stuck in too much water and is having a hard time controlling how much water is going in, that can actually cause the cell serious damage. So we're going to take a look at what kind of things can happen when osmosis kind of goes out of control. Before we start that, let's do a quick review of solution. So remember, a solution is a mixture of two things called a solute and a solvent. The solvent is the molecule that is going to dissolve something called the solute. In living things, water is the universal solvent. So remember, we are 70% water. That's because all the chemicals and the reactions that happen in our body happen in water. It's really good at dissolving stuff. The solute is the chemical that is going to get dissolved into the solvent. So for example, if you were making salt water, salt is a solute that gets dissolved in the solvent. Okay, so those two terms we're going to use, especially the word solute. So when we're talking about solutions in living things, we have a couple of different terms. A hypotonic solution is a solution that has lower concentration of solutes and higher concentrations of water than inside the cell. So if you have a cell, here's a cell, and you put it into a water solution, and there is more water outside the cell than inside, then you have put it into a hypotonic solution. Okay? What's going to happen is the water is going to move to the inside of the cell. There's more water outside than inside, so water is going to rush into the cell. What can actually occur when this happens is the cell can actually swell up, and if there's no cell wall, it can actually burst open. This is called cytolysis. Okay, so here you can see this animal cell, water is moving in because there's more water outside than inside, to the point where it gets really, really big and fat, and it actually could break open because it can't take any more water. Okay, so this can happen actually to people that try to drink too much water and then don't go to the bathroom for a really long time. People have died from that because this will happen in their kidneys and their whole organ systems will all shut down. Okay, so hypotonic, we've got more water on the outside than we do on the inside of the cell. Okay, so the water moves in. Okay, here are some red blood cells that are sitting in a hypotonic solution. They're fatter than they're supposed to be. The opposite of this is hypertonic. So in a hypertonic solution, you've got more solute in the solution that the cells are in than that are in the cell. So basically what's going to happen is you've got less water outside and more water inside, so the water wants to move out. Because remember, it's diffusion. They want to move to where there's more room. So the result of this is the water is actually going to move from the inside of the cell into the solution, and this can cause the cell to shrink. This is called plasmolysis. Okay, it's very easy to see this kind of thing happening in a plant cell. Here is a nice healthy plant cell, and if you put this, say it was like from a lettuce leaf or something, and you put it into salt water, all the cytoplasm, the cell membrane has all gotten squished in here. The cell wall stays the same because you can't change that, but everything else gets squished in, and when that happens, the cell can't function anymore. So this is what happens to your cells when you're dehydrated. They actually will start to shrink, and then they can't function anymore. And that's why you only can go a couple of days without water before enough of your cells get to this point where they're shrunken down, and then they can no longer function. Okay, so this is what one of our red blood cells would look like if it was shrinking because it was in a hypertonic solution. So water has been rushing out of the cell. Okay, so what we want is we want our solutions to be isotonic. So in an isotonic solution, the amount of solutes on the inside and outside are the same. Okay, that means the water is the same. So water is just going to move equally in both directions. And the cell remains the same size. So this is called dynamic equilibrium. Water is always moving, molecules are always moving, but the cell stays the same. Okay, so that's a nice healthy looking red blood cell right there, because it is in an isotonic solution. So this is what your red blood cells would look like when you were in homeostasis. Okay, so let's take a look at these cells and let's just see if you can figure out what kind of solutions these cells are in. So here we have an animal cell that is all shrunken down. Here's a plant cell where the cell membrane has pulled away from the cell wall. So that would be a hypertonic solution because the water has left the cell. Here we have some nice healthy looking blood cells. This is a nice healthy looking plant cell. That is an isotonic solution. And then over here, our red blood cells have gotten really fat because water has been rushing in. Here you can see the cell wall is bulging out. The vacuole is huge, got lots and lots of water in it, and that's because these cells were put into a hypotonic solution, which means there was more water outside than inside, so the water rushed in, filling the cell. All right, so how do organisms deal with all this osmosis that is happening all the time? We have various ways of dealing with it, and different organisms can do different things. Bacteria and plants have cell walls, and that prevents them from overexpanding. Okay, the pressure that is exerted on the cell wall is actually called turgor pressure, and that comes from the vacuole. So the more water the vacuole has, the more pressure you're going to see on the wall. And that's what actually makes a plant look nice and healthy. Now, if you overwater a plant, you actually can start to destroy some of the chloroplasts, and that's when the plant starts to turn yellow. Okay, but the cell wall protects them from their cells actually bursting. Their cells aren't going to burst because they have a cell wall. Okay, protists like a paramecium, they have a special type of vacuole called a contractile vacuole that they can actually pump water out of their bodies, out of their cells, using this vacuole. So when they start to take too much water in, it all goes into this vacuole and then they spit it out. And they actually can use that as a propulsion system so they can actually move themselves forward by pushing this water out. Saltwater fish. Ever wonder how something like a fish could live in salt water? They actually are able to pump the salt out of their body using special gills so they do not dehydrate. So they are actually getting rid of the salt and keeping the water, and that keeps them in homeostasis. Okay? And in us, our cells are bathed in blood. And then we have our kidneys, which help to keep our blood isotonic by either removing excess salt or excess water, depending on what needs to happen. So your kidneys need to be functioning all the time in order to keep your body in homeostasis. So animals have come up with a lot of different ways, or organisms have come up with a lot of different ways to deal with the fact that water is everywhere and it is constantly moving, and there's a lot of pressure put on their cells.